Let me introduce you to Bella. She is suffering from type 1 diabetes, which means that her body doesn't have the ability to produce the hormone insulin. So to help her treat diabetes, we will be producing insulin through genetic engineering today. The first step is to isolate the gene for insulin from a pancreatic cell. To do this, we remove the mRNA from the cell and use reverse transcriptase to produce a complementary single-stranded DNA, or cDNA. Secondly, we need to convert the cDNA into a double-stranded DNA using DNA polymerase. Now let's look at a magnified version to make things easier. With the help of a restriction enzyme, we will cut the gene coding for the production of insulin, shown in red. As you can see, the use of a restriction enzyme has generated sticky ends on each side of the gene coding for the production of insulin. Now, using the same restriction enzyme, we have to cut the plasmid that we isolated from a bacterium. And just like the gene coding for insulin, the bacterial plasmid has also produced sticky ends on each side where we have cut the gene. However, because we have used the same restriction enzyme on both genes, the sticky ends on the bacterial plasmid are complementary to the sticky ends on the gene coding for insulin. Now, this will allow the annealing of the DNA, creating hydrogen bonds in between the genes. However, the hydrogen bonds are only holding the fragments weakly. But to finally reform a continuous DNA molecule, we have to use DNA ligase. At this stage, we have now produced our recombinant DNA. What we have to do now with this recombinant plasmid is to insert it in a bacterial cell. As you can see, the plasmid also contained a marker gene, shown in pink. This is for antibiotic resistance. Therefore, the bacteria can be grown in culture medium. Apart from the recombinant plasmid, there were also other plasmids reproduced
transformed
with the wrong DNA that we don't want. Therefore, those cells that didn't obtain the recombinant plasmid with the marker gene will die, and the recombinant plasmid will grow. So once the bacteria has been incubated and grown, we then have to use a fermenter to allow the bacteria cells to multiply, and later we have to isolate the insulin from the bacteria and purify it to use it as medicine. So finally, Bella can use this insulin every day to treat her diabetes type 1 and keep her blood sugar levels normal.